Hello everyone! Today we are going to study the urinary system, and these are our learning objectives. Our urinary system consists of a pair of kidneys, a pair of ureters, a bladder, and a urethra. The urine is produced in our kidneys, and then it follows through the urinary tract, which consists of the organs that transport or store urine. So urine is produced in our kidneys, then it drains into the ureters, which are muscular tubes that through peristalsis carry urine from the kidneys to the urinary bladder. At the urinary bladder, urine is stored until it is released to the exterior of our body through the urethra. So we know our kidneys produce urine, but the question is, what does urine production mean to our body? And the answer is that through the formation of urine, our body eliminates ions, metabolic wastes, and extra water that it doesn't need. You need to keep in mind how urine is produced. And urine is produced from the filtration of our blood. Consequently, depending on how this filtration process is done, it can affect our body in different ways. And by filtering our blood, our kidneys can regulate the ion concentration in our blood plasma. And by doing that, our kidneys can stabilize our blood pH to the homeostatic levels, which is around 7.4. Also, when our blood passes through our kidneys, metabolic wastes are filtered out of our blood and become part of our urine. Metabolic wastes are biochemical wastes, right? And this is the waste formed from the biochemical reactions that happen in our body. And interestingly, most of the biochemical waste products are excreted out in our urine, not in our feces. Our feces are mostly food residues. And two biochemical waste examples that we have in our urine are urea and uric acid. Urea is the major organic waste product we produce, and it is produced from the breakdown of old proteins in our liver. The second major organic waste we produce is uric acid, which is formed by the breakdown of old nucleic acids. So when filtering our blood, our kidneys help us to eliminate these metabolic waste and ions, but at the same time, it helps us to conserve nutrients that do not need to be removed from our body. Also, by filtering our blood, our kidneys can regulate our blood pressure by regulating our blood volume. So, if we eliminate more water from our blood through more urine formation, then we can decrease the blood volume, which would decrease blood pressure. On the other hand, if we produce less urine, we retain more water, and with that we retain our blood volume, which could normalize or increase our blood pressure. So all these are functions of our urinary system, and they are all related to the blood filtration that happens in our kidneys. And just to give you an idea of how quick our kidneys are at their job, our kidneys filter and clean our entire blood plasma 60 times a day. And to do that, a huge amount of energy is spent. And this tells you how important our kidneys are to keep our body functioning properly. Now, besides these important functions of filtering out of our blood what we do not need, and at the same time preventing loss of valuable nutrients, our kidneys synthesize calcitriol, which is the active form of vitamin D. And our kidneys synthesize erythropoietin, which is a hormone involved with erythrocytes, red blood cells production. So what happens is that when our blood oxygen levels are low, some of our kidney cells release a hormone called erythropoietin, which stimulates our red bone marrow to make new red blood cells. Hence, we say that our kidneys are involved with erythrocytes, red blood cells production. So now that we know how important our kidneys are, let's talk about where they are located. And as you can see here, our kidneys are partially protected by some of the vertebral ribs. And we usually do not think that our kidneys are this high up. Our kidneys are outside of the peritoneal cavity. And remember when we covered digestive system, I told you about the peritoneal cavity. And I mentioned that our kidneys, ureters, and urinary bladder, they are all considered retroperitoneal. They were in the back of the peritoneal cavity. And when we do a dissection of a human body, if we open up the abdominal cavity and we remove the intestines, we would not be able to see clearly the kidneys there because they would be hidden behind the parietal peritoneum since they are behind the peritoneal cavity, right? So we would need to peel the parietal peritoneum out and then we would be able to see the kidneys. So our kidneys are right up against the posterior abdominal wall. And surrounding our kidneys, we have layers of connective tissue that help with stabilization and protection. Directly surrounding our kidneys, we have a fibrous capsule. Then we find fat called perinephric fat. And peri means nearby, right? Surrounding. And nephric stands for the little filtration units that we find in our kidneys that are called nephrons. So perinephric fat is the fat around the kidney. This fat around the kidney helps holding the kidney in position, and it helps protecting and insulating our kidneys because it serves as a little cushion against injury. Then after the perinephric fat, we have the renal fascia that helps anchoring our kidneys to the surrounding structures, such as the abdominal muscles that we have in the posterior body wall. Then you can see here we have some more fat over here and then the abdominal muscles and skin. This arrangement, all these layers help protecting our kidneys. And when someone loses a lot of weight and the amount of adipose tissue, the amount of fat to cushion our kidneys gets reduced, our kidneys become more vulnerable to traumatic injuries. So now you have an excuse to not lose weight because you want to protect your kidneys. Now, looking here at this diagram on the left, we can notice that our right kidney is a little lower than our left kidney. And the reason for that is because we have the liver, the right liver lobe, right above the right kidney. And if you recall, the liver is the biggest visceral organ we have, and the right lobe is huge and is located on the upper right side of our abdominal cavity. So because of our liver, our right kidney is pushed down a little bit, and then once again we can look symmetric. Another thing we can observe in this diagram is the inferior vena cava and the aorta, specifically the abdominal aorta. And branching off the abdominal aorta, we have the renal arteries. And each renal artery goes into each kidney, and then they branch up several times until capillaries are formed. And at the capillary level, excess water, excess ions, and metabolic wastes are filtered out of our blood. And this filtrate, after some other modifications, it turns into urine. And then the urine drains into this muscular tube called ureter, and urine goes down the ureter and it's collected in this muscular sac called urinary bladder. When the urinary bladder is full and it is appropriate to urinate, urine leaves the urinary bladder and travels through the urethra and gets out of our body. In this diagram, we have the anterior view of the abdominal pelvic cavity, and we can see once again that our right kidney is a little lower than our left kidney. And located right above our kidneys, we find the adrenal glands. And these glands are also known as suprarenal glands because they are located right above our kidneys. And these glands, the suprarenal glands or adrenal glands, they are endocrine glands that produce several hormones, including adrenaline and cortisol. Also in this diagram, we can see the hilum of our kidneys. And hilum is a word I mentioned before, right? We talked about the hilum of our lungs, which was the point, the area of entrance and exit of blood vessels, nerves, and also the entrance of the main bronchus. Now here in the kidneys, the hilum is the place where blood vessels and the renal pelvis will pass through. And when we look at this frontal section through the left kidney, we can have a better view of the renal pelvis passing through the hilum. Now let's analyze this diagram that we have here. We see that our kidney has an outer layer, which is called cortex. And we heard the word cortex before, right? For example, our cerebrum cortex, which is the most outside part of our cerebrum. So the cortex here is the outer layer of our kidney. Then we have the inner layer, which is called medulla. And the medulla has these triangular structures which are called renal pyramids. The tip of each renal pyramid is what we call renal papilla. And papilla means nipple. So apparently these little structures that we see here, they look like nipples. At the very tip of the renal papilla, we find the place where urine drips out. And then urine goes into a space called minor calyx. At the minor calyx, urine cannot be modified any further. Once urine gets into the minor calyx, that's it. All substances found in the urine present at the minor calyx will be eliminated from our body when we urinate. When minor calyx come together, they form what we call major calyx. And the major calyx drains into the renal pelvis, and that leads to the ureter that takes urine via peristalsis, so muscular waves of contractions force urine to go into the urinary bladder. Here we have the same frontal section of the left kidney that we saw before, but in this one we have a better view of the blood vessels. And I mentioned before that the renal artery branches off the abdominal aorta. And when the renal artery enters our kidney, it starts to branch out into different vessels until it branches so much that it turns into the capillaries that filter our blood. And this chart flow tells us the name of the vessels every time we have a new branch. Now, looking here at the kidney, we can observe that after the renal artery enters the kidney, it branches into the segmental artery. Then the segmental arteries branch into the interlobar arteries, which then makes this little turn that looks like an arch. And then they're called arcuate arteries. And then branching off the arcuate artery, we see these little branches that are like radiating into the cortex region of the kidney. And these vessels are called cortical radiate arteries. Now, from the cortical radiate artery, we have the afferent arterioles branching off. And the afferent arteriole branches into capillaries, which are the filtration system of our blood. These capillaries are called glomerular capillaries, and the group of capillaries is referred to as glomerulus. After the glomerulus, the group of glomerular capillaries, the efferent arteriole is what takes the filtered blood out, and then the efferent arteriole becomes capillaries again, which are named peritubular capillaries. But also, efferent arterioles can give rise to what we call vasa recta, and we'll talk about peritubular capillaries and vasa recta soon. Afterwards, these peritubular capillaries and the vasa recta will drain into venules. And then after the venules, we'll find the cortical radiate veins, then the arcuate veins, then the interlobar veins, and finally the renal vein that will drain into the inferior vena cava. Now, what I really want you to know is that renal arteries arrive at the kidney and they branch off several times. And then we have the afferent arteriole, and that's where we start. This afferent arteriole is what branches and becomes the glomerular capillaries, which collectively are called glomerulus. Leaving the glomerulus, we have the efferent arteriole, and the efferent arteriole branches, forming another capillary called peritubular capillary, and also they can form the vasa recta. After the peritubular capillaries and the vasa recta, blood drains into venules, and then blood eventually reaches the renal vein that drains into the inferior vena cava. And here we have magnified what happens when the afferent arteriole branches up and gives rise to the glomerular capillaries, which are collectively called glomerulus. And then after the glomerulus, the blood follows through the efferent arteriole. And looking here at the bottom diagram, you see that the efferent arteriole branches up again, giving rise to the peritubular capillaries. But also it can give rise to the vasa recta. And after blood passes through these peritubular capillaries and the vasa recta, blood will eventually drain into venules that will drain eventually into the renal vein that drains into the inferior vena cava. And this is what you need to know. So look here, the glomerular capillaries, we have these very funny looking cells surrounding it, because we can just see the capillaries in these little red sections that we see there. And surrounding the capillaries then, surrounding the endothelial cells that make up the capillary walls, we have these cells called podocytes. And podo means feet, right? And these cells, they look like they have little feet. And between the feet, between those projections, we see the endothelial cells that form the capillary, the glomerular capillaries. And through these little spaces, which are named filtration slits, is where blood plasma with substances in it will leak. And then whatever was filtered out of our blood ends up in this space called capsular space. And then it will follow through a system of tubules that we are seeing here, all these purple tubules. Now look at this. Surrounding the glomerular capillaries, we have this capsule that's called glomerular capsule or Bowman's capsule. And between the glomerular capsule and the glomerular capillaries, we have a space. And in this space, we will find whatever leaked out from the capillaries. So all that was filtered out of our blood will end up going into this capsular space. And then this filtrate will have no other option than to follow through the system of tubules that we see here. And that's why whatever that was leaked out of the glomerular capillaries is referred to filtrate or tubular fluid. So after the capsular space, this filtrate or tubular fluid will go into the proximal convoluted tubule. And the proximal convoluted tubule is called proximal because it's the one that's proximal to where the blood was filtered. And it is convoluted because it's all coiled up. So we have the filtrate going from the capsular space into the proximal convoluted tubule, and then it goes into what we call Nephron Loop, also known as loop of Henle. And the Nephron Loop has the descending limb and the ascending limb, and then after the ascending limb we have the distal convoluted tubule, which is the coiled up tubule that's distant from where the blood plasma was first filtered. Then after the distal convoluted tubule, the tubular fluid goes into the connecting tubule and then into the collecting duct. And as you can see here, the collecting duct collects tubular fluid from several connecting tubules. And then eventually this tubular fluid turns into urine, and that will be released into the minor calyx. Now, when we talk about the glomerulus together with the glomerular capsule, we say renal corpuscle. And when we are talking about the proximal convoluted tubule plus the nephron loop plus the distal convoluted tubule, we say renal tubule. And together, the renal corpuscle plus the renal tubule forms one nephron. And the nephron is the filtration unit of our kidneys. So here we have again the frontal section of our left kidney. And we can see the cortex and the medulla of our kidney. And we can see that we have two main types of nephrons. We have nephrons that are mostly located in the cortex of our kidneys, and we have nephrons that are mostly located in the medulla of our kidneys. And the nephron that's located mostly in the cortex is called cortical nephron. And the nephron that's mostly located in the medulla of our kidney is called juxtamedullary nephron. Now, let's look at this into more detail. We have the renal corpuscle of the cortical and the juxtamedullary nephrons. And both renal corpuscles are present in the cortical region, right? In the cortex of the kidney. And the same happens for the proximal convoluted tubule. Then we have the nephron loop. And the nephron loop is divided into descending limb and ascending limb. And that's where we see the biggest difference between the cortical and the juxtamedullary nephron, is the length of the nephron loop that dives into the medulla. So the juxtamedullary nephron goes deep into the medulla, and the cortical nephron doesn't go as deep. Then after the nephron loop, we have the distal convoluted tubule that connects with the collecting duct. Now, all that's filtered out of our blood goes into the capsular space, right? And then this fluid, this filtrate, is transported along the nephron tubules. And when it is transported along the nephron tubules, it gets modified because reabsorption or secretion happens along these tubules all the way until the collecting duct. And then some final modifications are done at the collecting duct level, and urine is finally formed. So the tubular fluid goes through this system of ducts and gets modified. And the main modification that happens at the level of the nephron loop is related to concentrating the urine. And now, since we know that the nephron loops are related to concentrating the urine, it becomes significant to differentiate the cortical nephron from the juxtamedullary nephrons, because the juxtamedullary nephrons, since they have a very huge nephron loop, they will be related to concentrating the urine. On the other hand, the cortical nephrons will not have that ability as much as the juxtamedullary does. So in our body, we have most of our nephrons are cortical nephrons because our urine is not that concentrated. We have a lot of water in our urine, right? So 85% of our nephrons are cortical nephrons. And just 15% of our nephrons are juxtamedullary nephrons because our urine is not that concentrated. Our urine is watery, which means that our urine has lots of water in it. And if we compare our urine to the urine of some mammals that live in the desert, you would see that those mammals, their urine is very viscous, is very like a paste, and that's because the amount of juxtamedullary nephrons they have is very high, and the juxtamedullary nephrons concentrate their urine so much that it turns into a paste. Okay, let's keep dissecting our nephron. So the nephron has four main parts, right? It starts with the renal corpuscle, and that leads into the proximal convoluted tubule, which is this coiled up tubule that's proximal to the renal corpuscle. Then the proximal convoluted tubule leads into the nephron loop, and there we have the descending limb and the ascending limb, and that leads into the distal convoluted tubule. That leads to the connecting tubule that connects with the collecting duct. Now, the nephron goes from the renal corpuscle to the end of the distal convoluted tubule. And we will go over every single part of the nephron and figure out what they do. There are three key mechanisms that nephrons use for urine formation. And the first one is filtration, which is the process of filtering or forcing fluid across the semi-permeable membrane. And filtration happens at the glomerular capillaries, right? And these capillaries are fenestrated capillaries. They have little pores. Remember, when we talked about capillaries, I mentioned that we had three different types of capillaries. We had the continuous capillaries, the fenestrated capillaries, and the sinusoidal capillaries. The fenestrated is the one that had little pores, and this is the one that we find here in the glomerular capillaries. So we have these little pores allowing filtration to happen. Surrounding the glomerular capillaries, we find cells called podocytes that I showed you in the previous slide. And these podocytes, they have little feet, little extensions, and between these little feet, we see the glomerular capillaries, these endothelial cells shining through. And those are the places where blood plasma will be filtered, so it will be able to leak out from the inside of the capillary. So filtration only happens at the glomerulus, at the glomerular capillaries. And what gets filtered out in the glomerular capillaries cannot be that big, right? Because the pores are very small. They are fenestrated capillaries. So we have small molecules such as water, ions, metabolic wastes, amino acids, and certain gases being able to pass through these little gaps. But cells are too big to cross these little gaps. So red blood cells, white blood cells, large proteins, they can't cross the filtration membrane. So they stay in the bloodstream. If red blood cells could pass through this filtration system, it would be normal to find blood in our urine. And that's not the case, right? We know something is wrong when we see blood in the urine. And that's because we know that red blood cells should not be filtered out of our blood when passing through these glomerular capillaries. So, under normal circumstances, just small substances are capable of crossing the glomerular capillaries. Now, after the filtration mechanism, we have the tubular reabsorption and then the tubular secretion happening. Tubular reabsorption means that we actually move substances from the tubular fluid back into our tissue fluid, which can then get reabsorbed back into our bloodstream. So reabsorption is the process of reabsorbing, moving fluid that is inside of the tubule back into the interstitial fluid. And then it goes into our bloodstream. So what would you expect to get reabsorbed? What would you not want to lose in the urine? And we do not want to lose in our urine any nutrients that were small enough to leak out of our glomerular capillaries. But we do not want metabolic wastes being reabsorbed, right? So we can conclude that reabsorption is a selective process. And we select what will be reabsorbed. And that involves transport proteins that use ATP to move molecules across the tubular membrane. And that's where we have lots and lots and lots of energy being spent to form urine the way it should be formed. Because we need to reabsorb all that was leaked out but should not leave our body. So we spend energy doing that. We use ATP to do that. Now, secretion is the opposite of reabsorption. So tubular secretion takes substance from our bloodstream and interstitial fluid and then puts them into the nephron. And what do you think our body would want to secrete into the nephron? And the answer is toxins. Toxins that are not small enough to leak out of the glomerular capillaries. Then our body spends energy to pump them into the nephron. And then that way it leaves our body as fast as it can because our body treats them as toxin. So filtration happens at the glomerular capillaries, and then whatever was filtered out of our blood goes into the proximal convoluted tubule. And at the proximal convoluted tubule is where most reabsorption happens. We have the reabsorption of 60% of the sodium and chloride ions that were leaked out. And we have reabsorption of some water as well. We have organic nutrients and plasma proteins being reabsorbed. And what allows the proximal convoluted tubule to reabsorb things very well is the fact that the cells that line the inside of the proximal convoluted tubule are simple cuboidal cells, like all other cells that make up the tubules. But these cells in the proximal convoluted tubule have lots and lots and lots of microvilli. And these microvilli increase the surface area for reabsorption. So lots of reabsorption happens here at the proximal convoluted tubule. So if any nutrients are leaked out by the filtration system, these nutrients are reabsorbed right away at the proximal convoluted tubule. Now, in the Nephron Loop, we get both reabsorption and secretion. And interestingly, in the descending limb of the Nephron Loop, we get reabsorption of water. And then in the ascending limb, we have the reabsorption of sodium and chloride ions. So the descending limb is named descending because as the filtrate flows through it, the filtrate goes from the cortex down, it descends into the medulla. And then we have the filtrate being forced back up, so ascending from the medulla back into the cortex, and that is the ascending limb. What's important to know about the descending limb is that the cells in the descending limb are only permeable to water, not to ions. So as fluid is being forced down the descending limb, only water is being reabsorbed at this point. So if only water is being reabsorbed, as the tubular fluid is going down the descending limb, that means that we are creating a smaller volume that's highly concentrated in ions. Because if you're removing water from the filtrate, what's left behind is a smaller volume with lots of solutes in it. And that's what we call concentrated. Now, when we get to the ascending limb, the cells that line the ascending limb are not permeable to water. They are only permeable to sodium and chloride ions. And then what happens is that in the cell membrane of these cells of the ascending limb, we find lots of sodium and chloride active pumps. So pumps that use ATP to actually pump sodium and chloride from the tubular fluid into the interstitial fluid, and that's called reabsorption. So when these ions are removed, we are actually deconcentrating the tubular fluid. So what that means is that the nephron loop concentrates and then deconcentrates the tubular fluid. And in reality, what happens is that water is pumped out of the descending limb because of the osmotic gradient created by the ions that are pumped out in the ascending limb. So by the end of the nephron loop, we have a smaller volume of diluted tubular fluid. Then in the distal convoluted tubule, what's interesting is that it is sensitive to lots of hormones. And hormones that influence the kidney, they will typically act at the level of the distal convoluted tubule or collecting duct. So, for example, if we talk about antidiuretic hormone, ADH, this hormone, it acts by increasing the amount of water channels in the distal convoluted tubule and collecting duct cells. And these water channels are called aquaporins. And by putting more water channels in the distal convoluted tubule and collecting duct cells, ADH, antidiuretic hormone, facilitates water movements through osmosis. So in the absence of ADH, water is not reabsorbed at the collecting duct. And when our blood osmolality is too high, so the amount of solute in our blood is too high, our pituitary gland senses it. And then it releases ADH. And then ADH stimulates the reabsorption of water in the cells that make up the distal convoluted tubule and collecting duct. So more water is retained in our body, less water is released in our urine. So what ends up happening is that we produce a small volume of urine that's highly concentrated. Another hormone that affects the distal convoluted tubule is aldosterone. And the way aldosterone works is that it aids in potassium secretion. And then what aldosterone does is to actually help remove potassium from our body and then secrete that potassium into our distal convoluted tubule. And at the same time, aldosterone aids in sodium reabsorption. So the point I'm trying to make is that the distal convoluted tubule can be affected by hormones. On the other hand, nephron loops are not really sensitive to hormones, and nephron loops will always do the same thing under normal circumstances. Now, the collecting duct, as I mentioned before, they are sensitive to antidiuretic hormone. And then they prevent diuresis, so it prevents water loss, right? So when the collecting duct is under the influence of antidiuretic hormone, it will help to produce a highly concentrated urine because it stimulates the absorption of water from the collecting duct. Now, my question is, can we call the fluid that we have inside the nephron urine? And the answer is no, we cannot call it urine, because what we have inside the nephron is the tubular fluid or filtrate that gets modified all the way until it gets into the collecting duct, and if there is presence of ADH, antidiuretic hormone, that hormone will act in the collecting duct and will keep modifying the tubular fluid. We can just call urine when it's out of the collecting duct, and that's when it reaches the minor calyx. So the first part where we can call urine is the minor calyx. Now, I mentioned previously that the efferent arteriole branches up and gives rise to the peritubular capillaries and vasa recta. And here you can see that these capillaries, they are strategically located surrounding the renal tubule. And there is a reason for them to be there that I call strategically located. These capillaries are there to pick up, to put back into the bloodstream all substances that were reabsorbed from the tubular fluid, as well as to secrete into the tubular fluid toxins and ions that our body wants to excrete. So, for example, all water that was reabsorbed from the descending limb and went into the interstitial fluid now goes into these capillaries and then is added to our blood volume. But now there is a difference between the cortical and the juxtamedullary nephrons. Remember, the juxtamedullary nephrons were the one that went deep into the medulla, and they have very deep, long nephron loops. And when we have these deep, long, straight nephron loops, we have the vasa recta. So we just find vasa recta in the juxtamedullary nephrons. And we do not find vasa recta in the cortical nephrons. In the cortical nephrons, we find the peritubular capillaries. So we find the peritubular capillaries surrounding the renal tubule of the cortical nephron, and we find peritubular capillaries surrounding the distal convoluted tubule and the proximal convoluted tubule of the juxtamedullary nephrons, but surrounding the nephron loop of the juxtamedullary nephrons, we have vasa recta. Now that we have a better understanding of what the nephron does, we can look at how it is organized in real life. And in real life, what happens is that the very end of the nephron, so the distal convoluted tubule, associates with the beginning of the nephron, which is the renal corpuscle. And this association forms a complex called Juxtaglomerular Complex. And the cells present in this Juxtaglomerular Complex, they are very important, and they aid in the process of measuring substances and blood pressure. So, making this Juxtaglomerular Complex, we have the macula densa cells, the juxtaglomerular cells, and the extraglomerular mesangial cells. The macula densa cells are found in the distal convoluted tubule, right next to the afferent arteriole. And these macula densa cells have chemoreceptors that sense the fluid inside of the distal convoluted tubule, and they measure the concentration of sodium and chloride present in the tubular fluid. And that's very important, because by measuring sodium and chloride present in our tubular fluid, these cells can understand what our nephron did, since we are looking at the end of the nephron. So if the sodium chloride content is high by the time the fluid gets to the end of the nephron, that suggests that the fluid moved too quickly through the nephron, and there was not enough time to reabsorb substances. Now, if the concentration of sodium chloride in the filtrate sensed by the macula densa cells is too low, that suggests that our tubular fluid moved through the nephron too slowly, which means we have too much time to reabsorb sodium and chloride. So the purpose of the macula densa cells is to measure sodium and chloride in the filtrate at the level of the distal convoluted tubule, and with that, indirectly understand how fast the tubular fluid is flowing, is moving through the nephron. Another type of cells that we find in the juxtaglomerular complex are named juxtaglomerular cells. And these cells are basically smooth muscle cells in the wall of the afferent arteriole. And their function is to effectively measure the pressure of the blood that's getting into the glomerular capillaries. These cells have mechanoreceptors, and they measure the stretch on the afferent arteriole. So if our afferent arteriole is really stretched, that suggests our blood pressure is high, and then these juxtaglomerular cells can actually contract to prevent damage to our glomerular capillaries. Because if our blood pressure is too high, that could potentially pop the glomerular capillaries. So the juxtaglomerular cells sense if there is too much stretch, and they can contract, and with that they vasoconstrict the afferent arteriole, and that reduces blood flow into the glomerular capillaries, which protects the glomerulus. On the other hand, if blood pressure is too low, the juxtaglomerular cells don't stretch enough, and then these cells release a hormone called renin. And renin is a hormone, but it is also an enzyme. And when renin gets released in the bloodstream, it converts angiotensinogen into angiotensin 1. And angiotensin 1 is converted into angiotensin 2 by ACE enzyme, which is in the capillaries of our lungs. Then angiotensin 2 has 4 main effects in our body, which are all related to increased blood pressure. Angiotensin 2 makes us thirsty, It causes the release of aldosterone, it causes the release of antidiuretic hormone, and it is a vasoconstrictor. So the overall function of renin is to increase our blood pressure, and that's all I needed to know. That when the juxtaglomerular cells do not stretch enough, they release renin, and renin leads to the increase of our blood pressure. Now, the last cells we have in the juxtaglomerular complex are called extraglomerular mesangial cells, and these cells are messengers. They facilitate the communication between the macula densa cells and the juxtaglomerular cells. Now we know how urine is formed, and after it's formed, it goes into the minor calyx. Then after the minor calyx, it goes into the major calyx, then renal pelvis, and then it goes into the ureter. We have a pair of ureters, which are muscular tubes that originate at the renal pelvis and extend down and connect to the posterior wall of the urinary bladder, where they drain urine into the bladder. When we look at a transverse section of a ureter, we see that lining the lumen of the ureter, we have transitional epithelium. And if you recall, transitional epithelium is a type of stratified epithelium. We have several layers of cells, but these cells can stretch and recoil back to the original shape in order to adapt to the degree of distension needed. So we find transitional epithelium lining the lumen of the ureter and also lining the lumen of the urinary bladder that I will be talking soon. Then we have here a smooth muscle layer, and these smooth muscle fibers will move urine through peristaltic waves of contractions into the urinary bladder. So if you are lying down or if you are upside down, urine will go from the kidneys into the urinary bladder because urine is moved through peristaltic waves. And then after the smooth muscle layer, we have the adventitia layer, which is the outermost layer that basically anchors the ureter to the surrounding tissue. Now, the ureter is attached to the urinary bladder posteriorly, right? And that area is called trigone. And here we can see the trigone and the two orifices through which ureters deliver urine into the urinary bladder. And this trigone is like a funnel that funnels urine into the urethra. And it is clinically relevant because since it is at the base of the urinary bladder, very close to the urethra, it's a common place for infection to happen. Now, lining the lumen of our urinary bladder, we have transitional epithelium, just like the epithelium we had inside of the ureters. And there is also rugae, which are folds that allow for expansion, so our urinary bladder can stretch. Now, we see here the detrusor muscle, and this muscle forms the wall of our urinary bladder. And that's why we refer to our urinary bladder as a muscular sac that stores urine. When the detrusor muscle contracts, it squeezes the urinary bladder and forces urine into the urethra. Then urine is eliminated from our body. And we see here that continues with the detrusor muscle. At the junction between the urinary bladder and the urethra, we have the internal urethral sphincter. And when this sphincter relaxes, urine can go from the urinary bladder into the urethra. Now, since we see here the urethra passing through the prostate, we can conclude that this diagram is showing us the urethra of a male. And here we have a lateral view of the male urethra. And since it is very long, depending on where it's passing through, it receives a different name. So the segment of the urethra that's passing through the prostate gland receives the name of prostatic urethra. After that, right under the prostate gland, we have the external urethral sphincter. That little piece of urethra that's passing through the external urethral sphincter is called membranous urethra. Now, after the membranous urethra, we have the spongy urethra. And this is the longest section of the male urethra. And it goes through the corpus spongiosum, which is one of the two erectile tissues I will talk about when I cover the male reproductive system. So the spongy urethra goes all the way down and it ends at the external urethral orifice. Now, when we compare the male and the female urethra, we can easily notice that the male urethra is much longer than the female urethra. And that's one of the reasons why males have much lower incidence of urinary tract infections than women. And another very important fact is the one that I mentioned when we covered the digestive system, and that's related to the wiping issue that some women have. Some women, they wipe in the wrong direction. They wipe from back to front, and that can bring bacteria from the digestive system into the urinary system. Because the female urethra is very short, women become more susceptible to urinary tract infections because bacteria do not need to go too far to be able to get into the urinary bladder and cause problems. In men, because the urethra is very long, bacteria needs much more time, and this time that the bacteria needs to travel through the urethra, it gives enough time for the white blood cells to take care of them and prevent the infection. And besides that, it is believed that the prostate gland produces a secretion that actually prevents men from getting urinary tract infections. Another difference that we see between males and females' urethra is that in men, the urethra is shared between the urinary and the reproductive system. On the other hand, in females, the urethra is not shared with the reproductive system. So to break any kind of misunderstanding, women do not urinate from their vagina, even though a lot of people think that. So here we see the uterus, the place where the fetus grows, and then we can see that the vaginal canal is a different tube than the urethra. Now, in both males and females, the urethra extends from the bladder, and right at the junction of the bladder with the urethra, and continues with the detrusor muscle that makes the wall of our urinary bladder, we find the internal urethral sphincter. Both the detrusor muscle and the internal urethral sphincter are made of smooth muscle fibers. We do not have control over them. And then a little further down, the urethra passes through the external urethral sphincter. And then it keeps traveling until it reaches the external urethral orifice, which leads to the exterior of the body. The external urethral sphincter is made of skeletal muscle fibers, so we do have control over it. The detrusor muscle is smooth muscle fibers. We do not consciously control the contraction of our urinary bladder. And also made from smooth muscle fibers is the internal urethral sphincter. Now, urination is coordinated by a reflex that's called Micturition Reflex. And micturition is the clinical word for urination. So what happens is that when our bladder is full, the stretching of the bladder stretch baroreceptors that then send information to the spinal cord and then to our brain, and then we become aware that we need to urinate. This leads to the contraction of the detrusor muscle and relaxation of the internal urethral sphincter. When the smooth muscle fibers that make the detrusor muscle contract, they squeeze our urinary bladder, and then it forces urine out. And at the same time, the relaxation of the smooth muscle fibers that make the internal urethral sphincter leads to the opening of the internal urethral sphincter, and then urine from the urinary bladder can pass into the urethra. Now, the only reason we do not urinate when it's not appropriate is because we contract the skeletal muscle fibers that make up our external urethral sphincter. Because we do have conscious control over skeletal muscle fibers, right? So we keep squeezing the external urethral sphincter until it is appropriate to urinate. And that's more or less the same situation we have with the internal and external anal sphincters. So we have the internal urethral sphincter, which is made up of smooth muscle fibers relaxing, but we can squeeze the external urethral sphincter that's made of skeletal muscle fibers, and then we do not urinate when it's not appropriate. And with this, we finish the urinary system. Please let me know if you have any questions. Bye!